You see dirt on a counter, you wipe it, it looks shiny, and you probably think to yourself, it's clean. I mean, we all do it.
Right, yeah. It's human nature.
Exactly. We just rely on this visual, basically binary test of safety. But the next time you pump a bottle of 100% pure alcohol hand sanitizer, or like grab a household disinfecting wipe for your kitchen, you might actually be protecting the bacteria instead of killing it.
Which is a terrifying thought.
It really is. So welcome to Deep Dive. For you listening right now, we know you want to cut through the noise, the information overload, and just get straight to the facts. Today, our mission is to completely transform how you look at the cleaning products under your sink, the sanitizer in your pocket, and, the protocols keeping hospitals safe.
Yeah, we are decoding the everyday chemistry of chemical microbial control.
Right. Specifically, disinfectants and antiseptics. We've got a fascinating stack of microbiology notes and historical medical case studies today. So, okay, let's unpack this. Before we even get to the actual chemistry, we have to start with the vocabulary. Because, well, everyday language gets this so completely wrong.
Oh, absolutely.
And the wildest thing I realized in our sources is that the difference between a disinfectant and an antiseptic has absolutely nothing to do with what is actually inside the bottle.
No, not at all. It's entirely about the target. Like, we tend to throw words around like they're synonyms, right?
Yeah.
Disinfecting, sanitizing, sterilizing.
Right, like they all just mean clean.
Exactly.
Yeah.
But to a microbiologist, blurring those lines is like a mechanic confusing a steering wheel with the brakes. I mean, they serve totally distinct functions.
Okay, so break that down for us.
Sure. So disinfectants are chemical agents applied to inanimate objects and surfaces. Countertops, doorknobs, floors, hospital bed rails.
Just non-living stuff.
Right. Whereas antiseptics are applied to living tissue. That means your skin, mucous membranes, or like an open wound.
So it's the exact same weapon, just aimed at a totally different battlefield.
That's a great way to put it. Take a common chemical, like chlorhexidine.
Okay.
If I scrub a patient's arm with it before putting in an IV line, it's an antiseptic.
Right, because it's on the skin.
Exactly.
Yeah.
But if I take that exact same liquid, the same bottle, and use it to wipe down a stethoscope, it is functionally acting as a disinfectant.
That is wild. It's just context.
Yeah, and that distinction sits inside a much larger, very strict scale of terms that microbiologists use. Like at the very top of that scale, you have sterilization.
And that means, like, literally everything is dead.
Yes. The absolute, complete elimination of all microbial life, including, incredibly tough bacterial endospores.
Wow.
And you usually cannot achieve true sterilization with just liquid chemicals in a bottle. You need an autoclave, which uses high-pressure steam, or like ethylene oxide gas, or heavy radiation.
So it's basically the nuclear option. You are leaving absolutely zero survivors.
Right. And then below that absolute peak is disinfection, which kills most pathogens on non-living surfaces, but, not necessarily those really tough spores.
Okay.
Then comes antisepsis, which is just reducing the microbial load on living tissue to a level that prevents infection. And finally, at the very bottom is sanitization.
Which is just lowering the microbial count to what is officially considered, like, publicly safe, right?
Exactly.
Like the quick wipe down a waiter gives a restaurant table between customers. It's just a much lower bar than true disinfection.
Yeah, it really is. And if we connect this to the bigger picture, from a historical perspective, how we measure and regulate these categories has shifted dramatically.
Right, because didn't they used to just rate everything on one single scale?
Yeah, early 20th century science judged these chemicals by a really crude yardstick. So in 1903, the standard was something called the Radiel-Walker test. It basically evaluated a chemical's killing power against one very specific bacteria, the one that causes typhoid. And it just compared it directly to phenol. It assigned every single chemical a single score, known as a phenol coefficient.
So the old 1903 method was like giving every chemical a universal horsepower rating, like assuming that if an engine is really powerful, it must be good for absolutely everything.
That's exactly it. But a single number wildly oversimplifies reality.
Well, yeah, because killing typhoid floating in a nice, clean glass test tube is a completely different physical challenge than, say, killing a fungal spore buried in a layer of dried blood.
Precisely. And the scientific community eventually realized that universal rating was dangerously flawed. I mean, killing power varies enormously depending on the specific organism, the ambient temperature, contact time, and whether there's organic matter present.
You're right.
So in 1968, a hospital epidemiologist named Earl H. Spalding introduced a totally new framework. And this is actually the exact system the CDC still relies on today.
Oh, wow. So what was the system?
Spalding argued for a tiered, purpose-based system.
So Spaulding basically realized you don't need a Ferrari engine to power a lawnmower. We need to match the chemical to the specific mission.
That is a perfect analogy. Spaulding sorted all medical items into 3 risk categories.
Okay.
You have critical items, like surgical scalpels that enter sterile tissue or the bloodstream. Those require absolute sterilization.
Right, obviously.
Then semi-critical items, like endoscopes that touch mucous membranes but, don't penetrate tissue. Those require high-level disinfection. And finally, non-critical items, like a blood pressure cuff that only touches intact, healthy skin.
Yeah.
Those just require low to intermediate level disinfection.
That makes so much more sense, and it really explains why a hospital supply closet isn't just stocked with one massive vat of super disinfectant. It's stocked with dozens of highly specialized tools.
Exactly. You match the weapon to the exact level of threat.
But, knowing what these modern chemicals do and how they are categorized naturally raises the question of why we started using them in the first place. And the origin story of chemical microbial control is, it's literally written in tragedy.
It really is.
It brings us to Ignaz Semmelweis in 1847.
A truly pivotal and, frankly, heartbreaking figure in medical history. He was a Hungarian obstetrician working at Vienna General Hospital.
And he noticed something completely horrifying in the hospital's structure, right?
Yeah. So there were 2 separate maternity wards. One was staffed entirely by midwives, and the mortality rate from puerperal fever, which is childbed fever, was quite low.
Okay.
But the other ward was staffed by doctors and medical students, and the mortality rate there was hovering around 18%.
That is just insane. Nearly 1 in 5 women dying shortly after childbirth.
It was a crisis. But Semmelweis realized the crucial difference in their daily routines. The doctors were coming straight from performing autopsies in the morgue directly to delivering babies.
Barehanded. Because, gloves weren't even a medical standard yet.
Barehanded, exactly. Semmelweis hypothesized that what he called cadaverous particles were being carried on the doctor's hands from the corpses to the mothers.
Wait, but he didn't know about bacteria yet, did he?
No, not at all. Germ theory just didn't exist. He didn't know what a bacteria was. He just knew something invisible was being transferred.
Right.
So he instituted a strict, uncompromising rule: mandatory hand washing with a chlorinated lime solution, which is basically a crude bleach antiseptic, before anyone could enter the maternity ward.
And the results are just instantaneous, right? I mean, the mortality rate plummets from 18% to roughly 1 or 2%.
Yeah.
He solved it. He definitively saved them. Wait, so he drops the death rate to 1%, and instead of throwing him a parade, the hospital fires him.
Fired, ridiculed, and systematically ostracized by the entire medical establishment.
How does that even happen? He had the actual data proving he was saving lives.
Well, the rejection basically came down to 2 major factors. First, he lacked the mechanism. Louis Pasteur and Robert Koch wouldn't scientifically prove the existence of microscopic pathogens for another 10 to 20 years.
Oh, wow. So he was way ahead of his time.
Way ahead. Semmelweis couldn't explain the underlying biology of why his chlorinated lime wash worked. He had empirical data, but no foundational science to actually support it.
And the second factor?
Pure ego.
Ah.
Yeah. The undeniable implication of his data was that doctors, the esteemed healers of upper-class society, were actively killing their own patients with their dirty hands.
Oh, man.
The medical establishment of the time simply couldn't handle that cognitive dissonance. It was easier to reject the data than to accept the guilt.
That is rough.
Semmelweis was driven out of his profession and eventually suffered a nervous breakdown. And in a really cruel twist of historical irony, he died in an asylum from a septic infection.
Oh my gosh.
Today, psychologists and scientists actually use the term the Semmelweis reflex. It describes the reflexive, knee-jerk rejection of new evidence just because it contradicts established paradigms.
That is just deeply tragic. But it contrasts so sharply with the other founding figure in early microbial control, Joseph Lister. This is a bit later, right? Around 1865 to 1867.
Yes, Lister was a British surgeon, and he possessed the crucial advantage of timing. He had actually read Louis Pasteur's new work on fermentation and germ theory.
So he had the science.
Exactly. Lister took that theory and applied it directly to the operating room. He specifically focused on compound fracture surgeries where a broken bone actively pierces the skin.
Which back then was basically a death sentence. I mean, mortality was commonly around 40 to 50% from sepsis alone.
It was horrifying. But Lister started using carbolic acid, which we now know as phenol. He sprayed it in the air of the operating theater, he soaked the surgical instruments in it, and he soaked the wound dressings in it.
Wow.
And because he could explain the mechanism that he was actively destroying the microscopic germs Pasteur just identified, his methods actually caught on.
Right.
He published his dramatically successful results, and he is now officially remembered as the father of antiseptic surgery.
And for anyone wondering, yes, the mouthwash brand Listerine was absolutely named in his honor.
It was ours.
It started out as a surgical antiseptic before becoming a consumer product, which is just wild to think about.
Yeah, quite a pivot.
But let's look at the actual science. Lister and Semmelweis were utilizing blindly because Semmelweis proved you can save lives without understanding the mechanism. But Lister proved that understanding the mechanism is how you change the world.
That's a great point.
So how do these modern chemicals actually rip a microbe apart?
Well, chemical microbial control fundamentally comes down to destroying a microbe's essential architectural structures. You are attacking either the cell membrane, their structural proteins, or their DNA and RNA.
Okay.
And because different chemical classes attack different specific targets, their spectrum, meaning what they're physically capable of killing, varies widely.
Let's start with everyday household bleach, or chlorine. It's described in the sources as an indiscriminate oxidizer.
Yeah, it doesn't pick a specific target. Chlorine aggressively strips electrons from the microbe's cellular components, essentially causing the cell to just fall apart at a molecular level.
Wow, just completely shreds it.
Pretty much. It's
No.
incredibly fast, very cheap, and broad spectrum. Like during the 2014 Ebola outbreak in West Africa, health workers relied heavily on dilute chlorine solutions to spray down boots, gloves, and surfaces because it destroys so much so quickly.
But it has an Achilles heel, doesn't it?
A massive one. Chlorine is rapidly neutralized by organic matter.
What does that mean in practice?
If there is blood, feces, or even just food residue on a surface, the chlorine violently reacts with the organic matter instead of the pathogen. It is essentially consumed by the dirt before it ever reaches the bacteria.
So if you spray bleach on a dirty counter, you're basically just disinfecting the dirt, not the counter itself.
Exactly.
Okay, then we have things like chlorhexidine and quaternary ammonium compounds, or quats for short. These target the cell membrane, right?
They do. They disrupt the membrane, basically popping it and causing the cell's internal contents to leak out.
Ouch!
Yeah. Chlorhexidine is the absolute workhorse in hospitals for surgical skin prep because it has residual activity. Unlike alcohol, which vanishes in seconds, chlorhexidine binds to the skin and keeps killing new microbes for hours.
And quats are the stuff inside those household disinfecting wipes you buy at the grocery store.
Right.
But they have a major operational gap, right? They are incredibly weak against non-enveloped viruses like norovirus. And I really want to make sure we understand this. Why does a chemical that works so well on one virus fail so completely on another?
It comes entirely down to the physical armor of the virus. Quaternary ammonium compounds work by plopping the outer lipid layer like the fat bubble that surrounds many pathogens, including the flu and the virus that causes COVID-19.
Okay, so it targets the fat bubble.
Right. Once that fat bubble pops, the virus is destroyed.
Yeah.
But non-enveloped viruses like norovirus, they don't have that fat layer. They are just tough, naked protein shells. So the quats have no fat layer to pop. The chemical just bounces right off the protein shell.
So if there's a norovirus outbreak on a cruise ship or, say, in a daycare, wiping everything down with standard quat wipes literally won't save you.
No, we won't.
You have to switch back to a heavy hitter like chlorine that actively destroys the proteins.
That is correct. You have to match the mechanism to the armor.
Okay, this brings us to what might be the most counterintuitive piece of chemistry we need to discuss. The great alcohol paradox.
Oh, this is a good one.
Here is what completely broke my brain in the sources. If you use pure 100% alcohol on a counter, you are actually doing a worse job than if you diluted it with water to 70%.
It sounds wrong, but it's true.
Is using 100% alcohol like trying to sear a cold steak in a blazing hot pan? Like, you burn the outside instantly, creating this hard crust, but the inside of the meat is completely raw.
I love that analogy, and it's chemically spot on. It comes down to how alcohols actually kill. They denature proteins, meaning they force the proteins inside the bacteria to unfold and lose their structural integrity.
Okay.
But protein denaturation is a heavily hydration-dependent process.
Meaning it requires water to physically happen.
Exactly. If you use 100% anhydrous alcohol, it evaporates incredibly fast,
Right.
and it instantly coagulates the proteins on the very outer surface of the bacterial cell. It creates a hardened shell, just like your seared steak,
Wow.
and then the alcohol can no longer penetrate that hardened shell. So the metabolic machinery inside the cell survives. The bacteria just goes dormant.
That's crazy.
But if you add water, making it a 60 to 90% alcohol solution, the evaporation slows down. The water acts as a transport vehicle, allowing the alcohol to penetrate deep into the cell and slowly, thoroughly denature all the proteins from the inside out, destroying it completely.
Which is exactly why hand sanitizer is always formulated with a significant amount of water. I mean, our intuition screams, stronger concentration equals stronger kill, but microbiology proves otherwise.
Nature simply doesn't care about our intuition.
Clearly. So we have established that diluting alcohol perfectly is the key to penetrating the cell, and matching the chemical to the fat layer is how you pop a virus.
Right.
But what happens when the bacteria doesn't just resist the penetration, but actually starts treating your chemical weapon like a food source?
Yeah, it sounds like bad science fiction, but it's a terrifying reality inside modern hospitals. We are talking about contaminated antiseptics.
Hold on, I need to stop you right there. Are you saying a chemical manufactured for the sole purpose of killing bacteria can actually keep them alive? Like, how does a living, reproducing bacteria metabolically thrive inside a literal bottle of poison?
Well, certain bacteria, specifically Pseudomonas or Serratia, are incredibly resourceful. If an antiseptic, like benzalkonium chloride or even chlorhexidine, is stored improperly, sits open on a cart for weeks, or is formulated at slightly too low a concentration, it just stops being a lethal threat.
Ow.
These specific bacteria have evolved efflux pumps. It's basically molecular machinery that pumps the toxic chemical back out of their cells as fast as it comes in.
Oh, wow.
And once they establish that tolerance, they actually use the trace organic nutrients floating in the water of the solution to survive and reproduce.
So a nurse could reach for a skin prep swab, thinking it is completely sterile, and accidentally introduce a thriving colony of Pseudomonas directly into a patient's bloodstream.
Unfortunately, yes. That exact scenario has caused multiple documented hospital outbreaks. It's a profound lesson that an open bottle on a hospital cart is not a permanently sterile environment.
That is terrifying.
It is, and the limits of our chemicals become even more glaring when we talk about endospores.
Right, bacterial spores like C. diff or anthrax, everyday quat wipes don't even make them flinch.
Spores are like microscopic bunkers. Bacteria form them when they're under extreme environmental stress. They shut down all their metabolic activity, expel water, and build a nearly impenetrable, multi-layered armor of specialized proteins.
So they just wait it out.
Exactly.
Ugh.
Standard surface disinfectants just bounce off. If a hospital ward has a C. diff outbreak, they have to physically remove the standard quat wipes from the rooms and switch exclusively to highly toxic sporicidal chlorine agents.
Wow.
If a hurried staff member grabs the wrong product, the infection spreads to the next patient.
But even spores look weak compared to the ultimate edge case, the one that completely defies the rules of biology. Prions. I was trying to wrap my head around this. How do you kill something that isn't technically alive?
Prions force us to fundamentally redefine what sterile even means. I mean, prions aren't bacteria. They aren't viruses. They have no DNA or RNA.
So what are they?
They're simply misfolded proteins that cause fatal neurodegenerative diseases, like Creutzfeldt-Jakob disease, which is analogous to mad cow disease in humans.
And because they are just proteins with no genetic material to destroy, standard chemical attacks and heat don't work.
They are astoundingly immune to standard sterilization protocols. There have been tragic documented cases where expensive neurosurgical instruments were used on a patient with an undiagnosed prion disease.
Oh, no.
They were put through standard hospital autoclave sterilization and then used on a new patient who subsequently contracted the fatal disease.
That is deeply unsettling. The protocols completely fail.
It forced a total rewrite of surgical procedures. To reliably destroy prions on surgical tools, hospitals now have to use extended autoclave cycles, so subjecting the tools to massive heat and pressure for a much longer time, and combine that with soaking the instruments in extreme, highly concentrated sodium hydroxide or hypochlorite.
Just to be sure.
Right. It proves that disinfected and sterile are not absolute permanent states of being in the universe.
Yeah.
They're merely claims relative to a specific evolving biological threat.
Speaking of evolving biological threats, we also have to touch on what happens when humans overplay their hand. When regulators step in and say a chemical has actually become a public health hazard, like the 2016 triclosan ban.
Yeah. For decades, triclosan was in absolutely everything. Hand soaps, toothpaste, body wash, even cutting boards. It was marketed heavily to consumers as an essential antibacterial agent. But the FDA banned it from over-the-counter consumer hand and body washes.
And they didn't just ban it on a whim. The manufacturers completely failed to prove that washing your hands with triclosan soap kept you any healthier or prevented any more illness than just washing with plain old soap and water.
The lack of benefit was only half the story, though. The mechanism of the risk was the deciding factor.
What do you mean?
Well, when millions of people wash their hands with triclosan, millions of gallons of this antimicrobial chemical are flushed down the drain and into our water supply. It gets heavily diluted. So bacteria in the environment encounter a very weak, sublethal version of the chemical. It's not enough to kill them, but it is enough environmental stress to trigger their defensive mutations.
Oh, man.
Yeah, they survive, multiply, and pass on that resistance. We were essentially running a massive, uncontrolled experiment in breeding superbugs.
That's horrifying.
Combined with unresolved concerns about hormonal effects on humans, the FDA just decided the risk-to-benefit ratio was entirely broken.
It's a stark reminder that more antimicrobial chemical exposure is not automatically better for public health, which transitions perfectly into the everyday stakes for you, the listener. How does all this complex hospital-grade microbiology impact your daily life? Because especially in a post-COVID world, we are all functioning as, amateur infection control officers in our own homes.
Very true. And it starts with how we interpret the labels on the products we buy. We see antibacterial and disinfectant in the cleaning aisle and assume they are totally interchangeable.
Right.
But they are legally and scientifically very different.
Yeah, antibacterial on consumer products often just means the product causes some general reduction in bacteria. It's a pretty loose term. But disinfectant in the United States is a strict EPA-regulated pesticidal claim under a law called FIFRA.
Yes, the Federal Insecticide, Fungicide, and Rodenticide Act. To put the word disinfectant on a bottle, a company has to provide registered, scientifically rigorous efficacy data, proving it kills very specific pathogens, like the flu virus or salmonella, to a very high predetermined threshold.
So we look for the EPA registration, we have the right chemical, but earlier we talked about the organic soil trap with bleach. That exact trap applies to your kitchen too.
Oh, absolutely.
So spraying a high-grade EPA-registered disinfectant on a sticky, crumb-covered kitchen counter is essentially like trying to paint over a wall that is covered in thick mud. You aren't touching the wall, you're just painting the mud.
I love that. That mud is actually a biological shield. The organic matter, the grease, the food, the spilled milk. It physically blocks the chemical from reaching the surface and consumes its active ingredients before it ever reaches the bacteria.
So you're wasting the chemical.
Completely. The golden rule of clinical and food safety protocols is a two-step process. You must clean first with soap or detergent to remove the mud, and then you apply the disinfectant to kill what's left on the wall.
Two distinct, non-negotiable steps. And what about the other massive household habit hand sanitizer?
We rely on it heavily, but as we've seen, it has hard biological limits. As we discussed with the quats, alcohol fails against non-enveloped viruses like norovirus. It fails entirely against spores like C. diff, and crucially, it performs terribly on visibly soiled hands.
Because of the dirt.
Exactly. If your hands have physical dirt or grease on them, the alcohol cannot penetrate through that grime to reach the microbes. Soap and water work completely differently. The soap molecules surround the dirt and microbes, lift them off your skin, and the water physically washes them down the sink.
That makes total sense.
That is exactly why hospitals mandate both hand sanitizer protocols and traditional hand washing, depending on the specific situation.
I remember during the height of the COVID-19 pandemic, this invisible chemistry was suddenly front-page news. The EPA created List N to track exactly what products killed the virus, and we all saw wipes being rationed. But there was also incredibly dangerous confusion. I mean, the CDC literally had to issue public warnings telling people not to ingest or inject bleach.
Which is the ultimate tragic misapplication of a highly toxic surface-level disinfectant to delicate living human tissue.
It really is. It just shows how high the stakes are when we don't understand the mechanisms of the chemicals we're using. We have gone on quite a journey today. I mean, we started with Semmelweis washing his hands in the dark, mocked by his peers because he lacked the underlying science. We explored the deeply counterintuitive hydration science of why diluting alcohol actually makes it a better killer. We learned that an open bottle of antiseptic can literally evolve into a bacterial swimming pool. And we realized that cleaning your kitchen counter requires removing the mud before you deploy the chemical weapons.
And the takeaway for you, listening right now,
Yeah.
is that every single time you grab a disinfecting wipe or pump a bottle of hand sanitizer, you are making a highly consequential applied microbiology decision. You are
Exactly.
choosing a specific chemical concentration. You must factor in contact time, actually letting that surface stay visibly wet for the required minutes instead of wiping it dry immediately. And you're matching the chemical mechanism to the specific biological armor of the target.
Before we go, what is one last thread from this massive topic that we can leave the listener to chew on?
Well, today we have mostly talked about these chemicals attacking free-floating individual bacterial cells. But what happens when microbes stop floating and start building microscopic, protective, chemical-resistant cities on your countertops or inside the water pipes of your home?
Wait, microbial cities.
Yeah, these structures are called biofilms. The bacteria encase themselves in a tough, incredibly resilient chemical matrix. So the next time you see a stubborn pink slime ring in your shower or sink that bleach won't quite fix, ask yourself, are you fighting a single microbe? Or are you trying to siege an entire fortified microbial civilization?
Okay, I am never going to look at my bathroom sink the same way again. That brings us right back to where we started. The visual test, clean or dirty, is a complete illusion. The real battle is invisible, and it's happening every time you reach for the spray bottle. Thank you for joining us on this deep dive. Stay curious, keep questioning the labels on your products, and we'll see you next time.